Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
What if you could flip a metabolic switch inside every single cell in your body? I mean, a switch that tells it to burn fuel faster, but not because you ran an extra mile or something, but just by tweaking the efficiency of your internal power grid. Imagine the tiny powerhouses inside your cells suddenly deciding to work just a little bit harder.
It really does sound like the holy grail of metabolic science, doesn't it?
Yeah.
But for about a century, this whole concept has been completely overshadowed by a pretty terrible truth.
Yeah.
Which is that the most potent versions of this metabolic switch are, in fact, lethal poisons.
Okay, so that tension right there, that's what this whole deep dive is about.
Exactly. The tension between this huge therapeutic potential and a really dangerous history.
And we are diving into a really crucial piece of foundational research that I think offers a completely new way forward. This work comes from a collaboration between teams at the University of Technology, Sydney, and Memorial University of Newfoundland. It was published in the journal Chemical Science. And they focused on engineering what they call mitochondrial uncouplers. These are experimental molecules designed with just staggering precision to potentially treat things like obesity.
And our mission today is to take you right inside the cell, show you the molecular mechanism they figured out, and explain how these researchers found a way to get that calorie burning effect without the, fatal side effects.
So if you, the learner, want to understand if science has finally cracked the code for safe, effective weight management, you need to know the difference.
The difference between a molecule that causes a controlled leak and one that just blows the whole system up. And that difference, it turns out, comes down to where two tiny atoms are positioned on a chemical ring.
To really appreciate the breakthrough, we have to start small. I mean, at the scale of the mitochondria, the actual engine room of our metabolism.
That's right. When we talk about the powerhouses of the cell, we are talking about mitochondria. Their whole job is converting the energy that's stored in the food we eat, so glucose, fats, into the cell's usable chemical currency, adenosine triphosphate, or ATP.
And this conversion process is not simple. It's not just a one-step thing. It relies on creating this huge force differential, which is why I think the hydroelectric dam analogy is so perfect for visualizing it.
It's the best analogy. Think of the mitochondrial inner membrane as the massive concrete wall of a dam. Now, the process starts when you oxidize nutrients from food. That energy drives what we call the electron transport chain.
Okay, and that's a series of proteins.
A series of protein complexes, yes, embedded right there in the dam wall. And this chain, it acts like a really powerful series of pumps.
Pumps for what?
For protons. As electrons move down the chain, the energy they release is used to physically pump protons, which are just positive hydrogen ions, across that inner membrane.
So you're building up this huge reservoir of positive charge on one side of the dam wall. You're stacking the water up high behind the concrete.
Precisely. That steep concentration gradient of protons, plus the electrical charge difference it creates, that's called the proton motive force. That's the potential energy, the stored water.
And normally, that water wants to flow back down.
It has to relieve the pressure, but it can only go through one specific channel, one special gate in the dam. And that's the ATP synthase complex.
This is our turbine.
This is our turbine. As the protons rush back through that ATP synthase turbine, the energy from that flow, it spins the enzyme, and it generates that high-energy molecule, ATP. That's the electricity.
So that's how it's coupled. The flow of protons is coupled to making useful energy.
That's the one. So when we talk about a mitochondrial uncoupler, it's doing exactly what the name implies. It's disrupting that coupling. An uncoupler is a molecule that essentially makes the dam wall leaky. It acts like a shunt pipe that lets the protons bypass the turbine completely.
So instead of the potential energy driving the turbine to make ATP, the uncoupler creates another path, a leak.
A leak. And all that energy from the protons rushing back through, it doesn't become useful ATP.
Where does it go?
It's just lost. It's dissipated entirely as heat.
Okay, so that's the core mechanism. But the cell must react to that, right? It's losing energy.
It does. And this is the trick we want to exploit. When the cell senses that its proton gradient has dropped and that it hasn't made enough ATP, it doesn't just stop. It assumes it hasn't burned enough fuel.
So it panics and starts burning more.
It absolutely panics. It ramps up nutrient oxidation, forcing the electron transport chain to run faster and faster to try and restore that gradient. It just starts tearing through more fats, more glucose, all in a frantic effort to stabilize its energy supply.
And that sustained increase in burning calories, That's the metabolic boost.
That is the boost scientists are trying to harness.
What's so fascinating about this is that while this new research is just groundbreaking, the concept of using uncouplers isn't new at all. In fact, the history here is incredibly dark, and it's a really necessary part of this whole story.
Oh, that history is crucial. It gives you the context for the immense risk involved here. These compounds were first discovered about a century ago, and the early versions were, to be blunt, indiscriminate cellular poisons.
And the cautionary tale really begins with one chemical in particular, 2-bill-4-dinitrophenol, or DMP. Most people today might know it as a dangerous, illegal weight loss drug you find online, but its story starts in industrial chemistry.
The famous story comes from around World War I. DNP was a common chemical used in French munitions factories, in making dyes, explosives, that sort of thing. And researchers at the time noticed that factory workers who are exposed to DNP, they started losing a lot of weight, seemingly without any effort.
But this was not healthy weight loss. The scientific notes from the time confirmed these workers often developed these dangerously high persistent fevers, a condition called hyperthermia.
And tragically, some of those exposures were fatal. Eventually, scientists connected the dots and realized this chemical was interfering directly with the cell's core energy production.
And once that link was made, it was incredibly effective.
Oh, astonishingly so. It was briefly marketed in the 1930s as one of the very first real weight loss drugs. It worked because it acted as a full uncoupler. It created such a catastrophic, massive leak in the dam wall that the cell's metabolism just went into immediate, desperate overdrive.
But the danger wasn't just how fast you lost weight. It was in the mechanism of death, which goes right back to that energy being released as heat.
That's the critical point. Since all that energy from burning food is diverted away from making useful ATP and is just released as pure heat, the body's internal temperature can spiral out of control. Fast.
We have a very narrow temperature range we can survive in.
Very narrow. And if your mitochondria are essentially turning into tiny furnaces inside every cell, your body just cannot get rid of that heat fast enough. And that leads to fatal hyperthermia. You literally cook from the inside out.
So the drug was banned because the therapeutic window, the difference between a dose that works and a dose that kills you, was just frighteningly narrow.
Dangerously close is an understatement. With DNP, you had no way to ensure you were causing a gentle, manageable leak. You were just risking an explosive breach that collapsed the whole system.
So for the last, 90 years, the challenge has been monumental. How do you get that metabolic boost without the fatal side effects?
How do you create a molecule that acts like a subtle dimmer switch, not an on-off explosion trigger?
And that's really what the scientists in this new study were trying to solve. They were trying to engineer an intrinsic safety throttle right into the molecule itself.
Right. If we connect that failure of the past to the potential of the future, the goal isn't just to find a new uncoupler. It's to find an exclusively mild uncoupler. Those old compounds, DMP or another one called FCCP, they were just too potent. You couldn't control them.
I appreciate you not trying to say the full name for FCCP.
It's a mouthful.
But this is why the new research is so significant. The team wasn't looking for a modified DMP. They were trying to find a completely new class of molecules that could be precisely controlled.
Exactly. They synthesized a series of novel molecules based on a structure called arylamide-substituted fatty acids. Their entire method was built on the idea that they could engineer a physical constraint into the chemical structure itself. A constraint that would predetermine a slower rate of proton transport.
Which first required defining what mild actually means in a measurable biological way that avoids that DMP toxicity.
The paper lays out three non-negotiable characteristics for a successful mild uncoupler. First, it must only cause a partial reduction of the proton gradient, just a partial depolarization. You want the water level in the dam to drop a little, not empty the whole reservoir.
Okay, so a small controlled leak.
Second, this should only weakly stimulate respiration, so a modest, sustainable boost in calorie burning, not a frantic overdrive.
And the third characteristic is the absolute safety valve.
The third, and the most critical, is that it has to do all this without compromising ATP synthesis, or cell viability. The cell must be able to keep making its usable energy. The leak can't be bigger than the cell's ability to keep pumping.
This means they needed a compound that was intrinsically self-limiting. They didn't want a molecule that could be toxic at high doses. They wanted one that couldn't collapse the gradient, even if you threw a lot of it at the cell.
That is the elegant design principle here. So they created and tested a whole library of these new arylamide compounds. And the goal was to see how very subtle changes to their chemical architecture affected their behavior.
Specifically, where certain atoms sat on the aromatic ring part of the molecule.
And the results of this structural manipulation were just stark. It cleanly divided all the molecules they made into two camps, the old toxic camp and this new, promising, safe camp.
All based on the location of just those two atoms. So what happened with the ones that acted like DNP?
Those were what they called the 305 dis-substituted Arylamides. In their tests on cells, these behaved just like the classic toxic uncouplers. They rapidly and extensively depolarized the mitochondria, and they caused a significant sustained drop in ATP production.
A drop in ATP is the red flag. That's the definition of dangerous. If the cell can't make energy, it dies.
Precisely.
Yeah.
The data showed that one of these, compound 3B, it dropped ATP levels dramatically, and it reduced cell viability down to less than 36%. This is the pathway to fatal toxicity.
But the breakthrough came with the slightly shifted addresses, the other configuration.
It did. The 3-fast-4 dis-substituted Arylamides compounds 5B and 6B. These are the scientific cutoff. They hit all three hallmarks of mild uncoupling. They significantly increased cellular respiration, which shows that real boost in calorie burning. And they only partially depolarize the mitochondria.
And most critically, they passed the safety test.
They passed with flying colors. They showed no significant effect on ATP production over the entire 6-hour test. And toxicity assays confirmed there was zero evidence of cell death or membrane damage, even at the highest concentrations.
So just by moving the substitution pattern from the 3-cellar-for-5 position to the 3-4-for-4 position, they turned a potential poison into a promising therapeutic agent.
That level of control is a monumental achievement in chemical engineering.
Okay, this is where we have to dive deepest. Because that shift from 3-5 to the 3-4, it seems so tiny chemically, but the biological result is night and day. This change must be throttling the actual speed of the leak.
It is all about the rate of proton transport. That's the core question. Why do the 3 other 4 compounds cause a mild, manageable leak, while the 3-5 compounds cause a gradient collapsing flood?
Right. If the transport rate is too high, if the leak is too fast, it just collapses the whole system. No ATP toxic uncoupling like we saw with DNP.
Correct. But if the rate is slow enough, the electron transport chain can keep up. The turbines can still spin, maybe with a little less pressure, but ATP synthesis continues. A slower rate is the definition of manageable, mild uncoupling.
Okay, so let's zoom in on the physics of how these specific molecules move protons. They don't just drift across the membrane, right? There's a mechanism.
It's a very sophisticated mechanism. It involves what we call the protonophoric cycle and dimerization. To get a proton across the fatty, oily core of that inner membrane, 2 of these individual molecules have to find each other and self-assemble. They have to dimerize. They join together into a pair.
Why do they have to form a pair?
Because that pair, that dimer, is what makes the complex lipophilic enough, fat-soluble enough to actually move through the membrane. And the formation of this 2-molecule complex, this dimer, is the single rate-limiting step of the entire transport cycle.
So the stability of that tiny 2-molecule complex, that's the bottleneck. If the dimer is super stable, it shuttles protons really fast, creating a flood.
And if the dimer is unstable, or it forms poorly, the whole shuttling process is slowed right down. You get a gentle leak.
That's the intellectual leap they made.
It is. And to prove it, they use techniques like NMR titration to directly measure how well these molecules could dimerize, and they quantify the stability using something called dimerization constants.
Let's break down that data. What did the numbers show for the toxic ones?
The full uncouplers, the toxic 3-5 compounds, they had extremely high dimerization constants. The highest was over 9,000 inverse molar units. A high constant just means the molecules strongly attract each other and form very stable, long-lasting dimers.
They're highly optimized to create that powerful leak.
Exactly. And the mild, safe uncouplers, the 3-4 compounds.
What about them?
They showed significantly weaker dimerization constants, dropping down to around 2,300, a clear 4-fold difference in stability.
Which proved the hypothesis. The 354 molecules form weaker, less stable dimers, so the shunt pipe they build is just less robust. It keeps falling apart, which inherently limits the flow.
It's an elegant, self-regulating safety mechanism built right into the structure. The less stable the dimer, the slower the rate of proton transport, and the safer the compound.
This raises the final and maybe the most intriguing question of the mechanism. Why? Why does just moving those atoms from the 3-5 to the 3-4 position suddenly make the dimerization so much less stable? It seems like such a subtle change.
You need computational chemistry to really see it. The answer is in something called the dipole angle, and how that affects the hydrogen bonding that holds the dimer together. The models show that the precise substitution pattern changes the molecule's overall polarization, its electrical field, which is critical for forming those bonds.
So where the atoms are sitting affects how well a molecule can stick to its partner.
That's a great way to put it. The 3005 disubstituted arylamides, the toxic ones, they are structurally perfect for this job. They had smaller dipole angles relative to the bond axis, meaning their chemical structure was perfectly aligned to promote strong, stable dimerization.
They were geometrically optimized to be the strongest possible proton shuttle.
Correct. Whereas the 3,4 disubstituted arylamides, the mild ones, had greater dipole angles. This difference in their physical shape results in a slight misalignment. It makes the hydrogen bonding less efficient.
And that structural interference is what slows down the rate-limiting step.
It is. So the breakthrough isn't just finding these two specific compounds. The breakthrough is establishing a conceptual framework, a blueprint.
By manipulating the substitution pattern, chemists can now predictably fine-tune the proton transport rate. They can base the safety throttle right into the molecular design.
It completely changes the game. Before, research was often about finding a low subtoxic dose of a really potent agent. This new work provides the tools to design an agent that cannot be potent, regardless of the dose, because its mechanism is intrinsically limited by its own geometry.
Okay, so let's shift from the elegance of the chemistry to what this means for human health. The immediate goal is obviously obesity, a huge risk factor for diabetes, heart disease, cancer. But the power to gently manage how mitochondria work, that seems to have benefits that go way beyond just weight loss.
Absolutely. One of the biggest adjacent benefits of mild uncoupling is a documented reduction in oxidative stress inside the cell.
And oxidative stress is basically the rust of the cell, right? It's the damage caused by byproducts from an inefficient energy system.
That's right. When mitochondria are running flat out or are slightly impaired, they produce excess reactive oxygen species, or ROS, highly damaging free radicals. Mild uncoupling helps the system cycle faster and more cleanly. By partially depolarizing the membrane, the cells avoid the conditions that lead to excess ROS formation. This cleaning effect is arguably even more important than the weight loss itself.
So you're not just burning more calories, you're making the remaining energy production more efficient and less damaging to the cell's DNA, its proteins, everything.
Precisely. And this mechanism has led to some pretty profound suggestions about the potential for these molecules in anti-aging research and, critically, in neuroprotection.
Things like Alzheimer's and Parkinson's.
Exactly. Those diseases are increasingly linked to mitochondrial dysfunction in our neurons. If the brain cells can't produce enough clean energy, they start to fail.
The idea that we could use a molecule to gently improve energy output and reduce the oxidative burden in brain cells, that's a totally novel approach to treating dementia. It's addressing the engine failure, not just the symptoms.
And you can compare this potential treatment to current obesity drugs. Many of the most popular and effective ones require regular injections and can have significant side effects. A safe, orally active compound that targets the core engine of metabolism without DMP's toxicity could offer a huge public health advantage.
But we have to keep perspective for the listener. This work, as amazing as it is, it provides a framework, a blueprint for designing drugs. These specific compounds, 5B and 6B, they are not in clinical trials. This is still foundational science.
That context is vital, yes. But the field of therapeutic uncoupling is incredibly active right now. We know another agent, a DNP prodrug called HU6, is already in phase 2 clinical trials, so pharmaceutical confidence in the theory is high.
But this new research offers a philosophically better approach. HU6 still relies on delivering a known poison, just in a controlled way. These arylamides bypass that danger completely.
They avoid the need to control a poison in the first place by engineering an agent that is intrinsically mild from the ground up. It's a fundamental step forward. It moves the focus from managing dosage risk to eliminating intrinsic risk through structural precision.
It's such a powerful demonstration of how this seemingly obscure structural chemistry can unlock these massive biological problems. We started with the terrifying history of DMP, the full uncoupler that acts like an explosion in the cellular power grid.
And we end with the elegant potential of these 3,804 disubstituted arylamides, molecules that use a subtle structural flaw, that less efficient dimerization, to create a slow, manageable leak.
Allowing the cell to burn more fuel, increase metabolism, and reduce cellular stress, all while keeping its critical ATP supply online. The entire journey hinged on where those two atoms sat on a single ring.
The most profound takeaway for me is the sheer audacity, of the control involved. They prove that controlling the stability of a tiny molecular dimer is the conceptual key.
Which raises a crucial question for you, the learner, to think about as this research moves forward. Given that the stability of something as small as a dimer can potentially unlock new treatments for diseases as widespread as obesity and as complex as dementia, how much undiscovered medical potential is currently hiding in the subtle chemistry of molecules we might otherwise just dismiss as nearly right?
And even more broadly, what are the long-term health benefits of a constant, mild reduction of cellular stress? If we can gently clean up the mitochondrial environment over an entire lifetime, the implications for human health and quality of life are truly profound and still largely unexplored.
It's a reminder that sometimes the biggest breakthroughs really do hide in the smallest, most precise molecular details. That was a deep dive into the world of mild mitochondrial uncouplers and the new age of chemical safety engineering.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.